Yes, so welcome to day six session on molecular modeling methods and types. So before going to day six session, can we go ahead with the recap of the last session topics? So what exactly did we discuss the last session?
Network pharmacology, sir.
No, your voice is feeble. What exactly you mean?
The network pharmacology.
You are right. You are right. What other things we have discussed? We have discussed on what is system biology, what is system pharmacology, and then we also discussed on precision-based medicine. Very good. Then what else did we discuss? Can I ask you a few questions related to that? Give me yes, good. So now we have studied the types of omics in system biology. So types of omics in systems biology. This is my first question. What are the types of omics we have discussed? Genomics, lysosomics, proteomics. Proteomics is missing. Good. Proteomics. Then already I mentioned like interactomics. Interactomics. Then metabolomics. Very good. So this is how we have discussed. Now the next is uh, yeah, with databases. What are the network databases? Network pharmacology databases we have discussed. What are the network pharmacology databases? So PPA interaction databases we have discussed. We have discussed one is string DB, and okay, network pharmacology we discussed herb impact. Very good. So you Vishnu and Samuel is answering very good interaction. You have a very good interaction. Now let me take you to the next thing. PPA interaction databases. What are the different PPA interaction databases we have discussed? Whether we have PPA interaction in Hub as well as in Impact, or we have discussed, give me some examples for PPA interaction databases. Can we explore more on the databases initially, and then we go to the use of Cytoscape? Very good. But network analyzed, okay, it's a kind of a gene interaction databases. Okay, so network analysis is basically about the gene interactions, and Cytoscape is the visualizer. So if you want to visualize the gene interaction, protein interaction, we can use this offline tool. So we can use the gene at Patchak. Patchak is also similar to Cytoscape. String TB. So this is the answer I'm anticipating. Other than string TB, what else did we have find for PPA interactions? Can I take you to the some websites and provide you more examples with PPA interactions? Now I take you inside. So for knowing about the PPA interactions, okay, we can also use this database called UniProt. UniProt refers Universal Protein Database. So this is one of the important biological database where we find full annotation of a protein. If we don't know anything about a protein, and but if you want to do a research in your UG or in PG or PhD, obviously this database gives more detail up to PhD information. Okay, now let me take you to annotating a detail of a protein. If I want to annotate a protein detail, that is a trap one. From beginning, I was explaining you about this target receptor. I'm not changing that. I'm taking you to the trap one. So trap one is a heat shock protein 75. It's a mitochondrial kind of a protein. Now I'm going to take you inside the trap one. So I just gone inside this trap one, and Uniprot. We have Uniprot knowledge base, Universal Protein Knowledge Base. This is the most important database present in Uniprot, where you can able to find fully annotation of the protein. What do you mean by annotation? If you don't know anything about a protein, if you want to know about the function of a protein, features, especially the binding sites of the protein, and then if you want to go across the gene ontology information, like what are the different interactions of the protein, what is the biological function of this protein, what are the so all this information, okay, and then enzymes information related to this protein, pathway databases are related to this protein. So it links also with the databases, and then we can see proteome database. Okay, so this is the largest proteome database you can find in UniProt. In UniProt, we have a proteomes, a separate database. Okay, if you want to explore on the set of related proteins of a specific organism, one can able to go through this UniProt proteomes. This UniProt, Universal Protein Database, present in London. This is governed by UK. Okay, so this is an European kind of a database, and here you can able to see also PPA interactions and disease variants. So what gene is responsible, what position of an amino acid is responsible for disease. So that information and single nucleotide polymorphism. So all this data is also interlinked. So organism specific databases, whatever I mentioned you on yesterday, DG network. So this is the DG network ID. If you want to explore on this trap one related kind of diseases, okay, in the network, so we can able to link. So all these are hyperlinks. If you click it, directly take you to that. So this is an organism specific databases. So in DG network for the trap one, we got this information. Okay, so I just show you what exactly we got. So we got around 10,131 information related to trap one. Okay, so like what disease? It is associated with this much number of diseases. Like it associates with diabetes mellitus. It's relate with the hypodysplasia, osteoarthritis, stomach neoplasms, breast cancer, so renal cell carcinoma. So like this tryptophan is an important target for many diseases. We can also see, we can download the TSV file, and after downloading the TSV file, we can open in the cytoscape. We can see the disease networks. So all this information you can able to find even in this DG network, which is related to diseases. Like however I mentioned about the DG network, okay, then we also have MIM, open targets, form GKP, malakas. Like this, we can also see the chemistry, like what are the drugs used for as a trap-1 inhibitor, and how to download these molecules. So this information we can able to take from the drug central database, guide to pharmacology, CHEMDL. So we can able to fetch the trap-1 inhibitor information from this UniProt. Okay, so if you want to do any research, no need to explore the data in any other websites. No need to even search through a specific literatures. Even the literatures are linked with this UniProt. So one day I'm calling this as a fully annotation database. We get full details of the specific protein or specific target, whatever we are searching. Okay, so first exploring this, even though NCBI protein is the largest protein sequence database, well, here, even if you have few proteins, we have full details for that few proteins. Okay, and we have proteomic databases separately. These are a few proteomic databases, and these are the unique identifiers where of the trap one, where it can directly take you to that specific protein, and 2D gel databases, post-translational modification databases, gene expression databases, organic specific databases. So we have plenty of databases. So protein-protein interaction. This is the question I asked you just now, isn't it? We just discussed on string. There is also one database called BioGrid, which is, of course, a largest database for protein-protein interactions. Like this, the fun cook, really it looks funny. The protein-protein interactions looks funny, but it's a kind of a good interaction. But you remember, you can't able to take as such. I mean, whatever data you take from this kind of a databases, you can't able to use this kind of an output directly to the publications. Okay, so why? Because we have data privacy. So one thing we can do, we can download the data in the form of .tsv, and we can open in Cytoscape. We can have some kind of color change, and we can do something based on the filtrations. After filtering the data and after incorporating the color change, we can use the data for publications. So this is how, okay, so how we can able to use this data for journal publications. So BioGrid. So you can able to see in this biogrid for the trap one we have around 486 protein genes are connected, okay, or connected with it, and publications we see around 162 publications are there for trap one. So we have plenty of information. Okay, don't simply restrict up to searching even the literature from the Google. Search through this kind of a websites where you get more information. Also, disease information. Like if you want to check with the diseases, so even this is the best website. Okay, to search for. And not only that, if you see this, we have plenty of data sets available. See, according to this thing, that trap one, we have an interactor called HSPA9. If you want to know more about HSPA9, you can click inside and you can get more details on that. But one thing you can see, the HSPA9 and TRAP1 is also going to interact with few proteins. Based on the protein-protein interaction, if you want to check, you can also check this physical interactions inside. Okay, you can click view and can go through that. Like Homo sapiens, you are also getting for different species like SARS-CoV-2. Okay, so how these protein-protein interactions are developed? If I click this view, okay, where we have four kind of data. Okay, the four data, they have captured from the two data captured from affinity capture mass spectrometry, and the other two data from the proximity labeled mass spectrometry. So these are the scientists they work, and related to curation data. So click this biogrid. So you'll be getting the specific publication, the other details, how this is linked with the trap one. Okay, so this information you can get. So like this, you can see interlinked kind of a databases. Also, we have CRISPR database, OMM database, Vega, Entrus. Entrus, Entrus is linked with, Entrus is the browser present in NCBI. I have taken you to the NCBI, isn't it? Do you remember how the NCBI website looks like? So ncba.nlm.nih.gov, isn't it? For the newcomers, you can try even in Google type NCB, and or else you type the entire URL, get inside. So whatever we got inside this kind of a desktop, okay, kind of a window of an NCBI, this is what we call it as an interest. If you want to link to the interest, okay, so there is also an interest, and this is an interest gene. It means that whatever you can able to search in this gene, okay, the trap one. If you search for a trap one, okay, that's what it's going to find from there. Okay, so like this, we have plenty of databases interlinked with the one gene, and this is a funcoop database. See how it looks like. Okay, so in funcoop database, this trap one is linked with many kind of proteins, a chit chat kind of emotions. You can also, so only they, I think they named as a funcoop, but you get more information of in this funcoop, that is protein-protein interaction database. So here, what exactly I can find? See, the trap one linked with proteins. What are the different proteins linked with the trap one? First, they've shown like a GRPL. So if I keep there, you can able to see this is getting highlighted. If I take it to this chain, and obviously can see what are the interacting genes of a GRPE1. So these are the genes. Okay, so you can able to find this chain, this chain. So this is a kind of a heat map, and how much it is interacting based on the intensity of color. You can able to see the kind of interaction. Okay, of course, GRP, the glucose regulated protein. So GRP stands for glucose regulated protein. Of course, this is one of the heat shock protein uh, isoform, and this also having a good interaction. Similarly, we can find plenty of proteins. We also have green and red based on positive regulation, negative regulation. So this information can also be found out in this fun code. As I illustrated you on yesterday, do you remember what I illustrated you on yesterday? I have taken you to the string TV. You remember in string TV, I have taken you and just start explaining that you can also, if you initially if you get 10 interactions, if you want to increase the interactions to 50, we have that information in the settings. Okay, so first initially I'm searching for a target protein inside the search, in the default search, and then I'm going to the Homo sapiens in defaultly, I'm searching for the human trap one. Now I got a trap one linked with 10 proteins in string TB database. Okay, so later if you want to increase the number of interactions, clicking the settings, instead of 10 interactors you can increase to 50, or if you want to customize the value up to 100 interactors, that is 100 interacting proteins with hub target protein trap one, we can able to give the custom value also. Okay, so maximum interactors, if you want 100 interactors, I just give the 100. And if you want to enable bubble design, you can select this. This also looks fine. So now see how it looks like initially, and I know I'm going to update it. So after updation, okay, so you can able to see nearly 100 proteins. So you can see the top 100 proteins which is interacting with the trap. So this is how we tried working on the system biology approach on the ovarian cancer, kidney cancer. You remember I sent you the articles. So the Scopus indexed and Sci indexed journal papers. So this is how we can able to find the interacting gene information. To know more about how this gene information we got, so we can able to see from the viewers. Inside the viewers, we have experiments. So see co-purification, co-crystallization, yeast to hybrid, genetic interactions. These are responsible. Okay, these immunotechnology experiments are used to create this protein-protein interactions. So in protein-protein interaction, we have to remember two important points. The first most important point is whether your gene is going to positively regulate or negatively regulate with the target gene. If trap1 is the major protein, so see this also looks like bubble. I changed it as bubble format, isn't it? So earlier it was not like a bubble, but now it looks like a bubble. Okay, so the trap one is going to interact with the top 100 proteins, isn't it? According to the settings, however we search. So this kind of a data, whatever we got, okay, so we can able to first check whether it's a positive regulation or negative regulation. Positive regulation denoted by red color, negative regulation by green color. So through the colors we can able to find. So one data is like this, and the second data we can able to find the length of the edge. The length of the edge. You remember, according to the graph theory, I told you in protein-protein interactions, whatever we keep proteins, we call this as nodes. If these bubbles, each and every bubble is a node, here at present we have 101 nodes. 101 nodes are connected with edges. This trap one is closely the edge of trap one to HSP90 alpha, HSP90 beta is less. So it's going to have a close interaction. This interaction is more close. The trap one to HSP90 alpha and HSP90 beta. But if you see few proteins, for example, TNFR, tumor necrosis factor receptor, they have a very long edge. So the interaction is very less. When compared to the heat shock protein 90 alpha and beta, okay, this interaction is less because of edge length. So like this, these are the major two data we can able to find in protein-protein interaction. So many scientists have worked on these experiments, isn't it? Now I'm just clicking this particular thing, okay, immunotechnology thing. Okay, so how many experiments are done and how many data sets are available. Okay, all these things we can able to see from this information. See, plenty of scientists. Okay, so group of scientists work on these kind of a data, and if you want to work once again, there is no need. We have plenty of data, so we can start from here, and then we can try to come up with a kind of a drug. Okay, so we have plenty of proteomics data at present. So see here, this is from one scientist. According to this scientist, the trap1 is one of the interacting protein of 1522 proteins. Okay, this assay was done through is performed in protein-protein interaction intact assay, anti-taq immunoprecipitation assay. So these are the assays they have used to check how the trapon is interacting with the 1522 proteins. And this is a different data by a different scientist. So here it is 1521, and here in this case 1018. Like this, we have plenty of data available for trap1 alone. Okay, and even the data is truncated for 10 items. I don't know how many data sets are available. Each and every combination of the gene data, we call that as data set. The last two, you can see the less numbers, 295 proteins, 238 proteins. You remember at present what is how I have searched? In StringDB, I have searched for 100 interacting proteins, top 100. So whatever 100 is top 100. Okay. So top 100 proteins are generated from this 1522 proteins. So this is how we have to find this information, and even what genetic or what species they have used for genetic uh, combination. All this information they are provided here. Okay, so in like however you find full information of a protein in Uniprot, to know about the protein interaction, fully annotation of protein interaction, one can go to the string DB. The string DB is the largest PPI interaction database, but we also have few databases like FunCoop and whatever I showed you here, FunCoop database of interacting proteins. This is what TIP, and similarly Mint, Intact. So these are protein interaction databases. Chemistry, if you want to check the binding sites of the specific target, we have it in binding TB. Okay, and similarly RNA act for RNA interactions of that specific trap one. We also have PDB structures. We have plenty of PDB IDs for the trap one based on the specific localization regions. If you want to focus on binding sites, or if you want to go with high resolution protein, 3.06, I told you it's a low resolution. If only if you work with high resolution, that is below 2 angstrom, you get more precise results. Okay, so even selecting the PDB ID is most important to check how precisely you are working on can be on molecular talking or MD simulation like that. Okay, so these databases, whatever I showed, Uniprot, StringDB, so this gives you a sufficient knowledge, okay, to work on system biology. So system biology is majorly you find these databases, we use it rarely. Okay, and other than that, we also go with other kind of a databases. Okay, so if you want to find this information, explore more on this Uniprot database. Now let me take you to the next thing. Like yesterday, you remember, I have downloaded a kind of PPI interaction thing from StringDB. So how to download the PPI interaction from StringDB? You want to show, make me show once again. So if you want to download the data, okay, so you can go to the exports. Inside the exports, you can click this download. If I click download, yesterday I think I have searched for 50 interacting proteins, but now I just change it to 100. If I click this download, you can able to get 100 interacting proteins. Okay, so 100 interacting protein data, you can able to find it here. So here CADVD means, so this is a short, but this is what 100 interactors, isn't it? I type like 100 interactors. So whatever I just downloaded like 100 interactors, we have to open this it in Cytoscape. So for opening the Cytoscape, okay, I've already installed this Cytoscape. Cytoscape is the free visualizer for all the types of networks. So the icon looks like this. So simply click this Cytoscape. It also occupies very less space. As I mentioned you on yesterday, in Cytoscape, we have plenty of apps. Of course, this is a Java enabled kind of a visualizer. We have plenty of apps. So whether you want to open DNA protein interaction, RNA protein interaction, we want to analyze any kind of filtering of data. So for each and every function, we have to install a specific apps, and then we can able to work on. Okay, the latest version is 3.10.4, but I have 3.8.1. It's asking whether you want to download and update this. No, I don't want. I simply want to use the whole thing. So I click close. So I just opened and showing you the visualizer. Okay, this is the window of a cytoscape. And if you want to open a specific colorectal cancer kind of a targets, so these are some of the demo files already present inside. So I just click the demo file. You can see this interaction of protein in colorectal cancer. Now let me take you to our file, whatever we have downloaded. If I want to open the file, whatever I have downloaded, okay, import network from the file system. So in the file system, I go back. So it's there in the CADVD. Presently, if I want to open 100 molecules, so now we can able to see this network in the form of table. So before opening it in the Cytoscape, I can also show you how this .tsv looks like in Excel. You want to open it in Excel, okay? So first open the Excel, Microsoft Excel or any Excel, whatever you have in your Windows or Linux. Then after opening this Linux, okay, you just drag and drop, or you just open, okay. You just have to open from the Excel sheet, okay. And now I'm just dragging and dropping here. Let me see. If I have a problem, okay, we got it now. See. So after dragging and dropping, we can get able to get this data. So wrap text. So you can able to see. I just zoom in and show you. So these are the data I can find. So in node 1, AHSA1. In node 2, we have a different protein interaction. So we have around 1500 interactions, isn't it? 1544, but we ask for 100, top 100. But the top 100, we can see each and every gene interacting with the other genes. Okay, so node 1, if is AHSA1, AHSA1 protein alone is interacting with 42 proteins. This alone is interacting with, I think, 41 proteins. 41 proteins. Similarly, you can see the next protein. So we have 100 into something. Like we have more than 1000 interactions. We have 100 proteins, but we have more than 1000 interactions according to this. So this is how the data, whatever data we downloaded from string DB looks like. Okay, these are the coordinate data. So now I'm closing this. So I'm closing this. I'm not going to save anything. Now whatever I don't say. So I just closing the Excel sheet. Now whatever data I open in the Excel sheet, the similar kind of a data you can able to see when you try to load the kind of the TSV data in Cytoscape. Now simply click OK. So after loading this, you can able to see this. Okay. So here you can able to see a plenty of proteins or interacting in the or you can in this protein interaction network. So later you can use the style, and you can change the style of the colors, and you can change the you can also use the filters, and based on the filters you can color the proteins. Okay. And there is one app called CytoHubba. If you want to calculate node scores using this CytoHubba, use this CytoHubba. So for installing these kind of apps, you can use this app store. Okay, use the app store. Whatever app you want to install, defaultly this app you can't able to find. You find only the network style filter annotation alone. So whatever you can find, network style filter annotation, this you can able to find in default Cytoscape. But if you want to use other kind of apps like a Cytohub M-Code, so you have to install from this app store. So for installed apps from file or show app store, from this you can able to install this. If I click show app store, you get many kind of an apps. Whatever app you want to install, you can select it and you can able to install it. I've already installed the site up. Now I'm taking it to the site up. So here in this, it's asking you to calculate node scores. So just click calculate node scores. Now it's asking you what nodes you are interested in. 3 nodes are generated. If you're interested on node A, we can select the node A. Or if you want to get the top 10 nodes ranked by different kind of parameters like MCC, TMNC, degree. Okay, so based on degree, if I want to know rank the node, so I select the degree and type top 10. Now yeah, I'm going to click submit. Okay, so it takes some time. Immediately won't get the result. So or else you can also select whatever nodes you are interested in, and you can able to check this information. Okay, so it's to know more about how to use this kind of a site or hub and other kind of a thing. Okay, so we can able to learn from the help. In the help, we have video demos. So it linked with the YouTube, and you can get so many kind of videos to how to use each and every app of the Cytoscape. Plenty of videos are available inside. So it adds to the YouTube videos. Similarly, you can able to see the tool. You can go with the tutorials. We have already existing files. We can work on it. But I suggest you to use even user manual. In user manual, you can find all the packages. I mean, all the kind of an apps and all the kind of functions what we can able to use. This is what the extensive API of the Cytoscape. Okay, so one should all try to use this kind of information from the help. Okay, to know more about this. Okay, so just I showed you that kind of the visualizer called Cytoscape, and I was explaining about PPI interactions. Like PPI interactions, you try with the disease gene networks, you try with the intact interactions, and other interactions also. Now, let me take you to the today's session. Today's session, I'm going to take you on to the molecular modeling methods and types. So, molecular modeling, you remember, I've already taken on the protein modeling, macromolecular modeling, but we also have small molecules. So molecular modeling cannot be of two types. It can be a small molecule modeling or macromolecular modeling. But first of all, what do you infer from this molecular modeling? Molecular modeling encompasses um, so molecular modeling encompasses all methods, theoretical and computational, used to model or mimic the behavior of molecules. If you want to check, okay, how the molecules, like gaseous molecules packed in the cylinder, whether it's for fire, it's going to exhibit the Brownian motion or not. So to check what is the whether this molecule is going to move or not, the dynamics, whatever we measure. So all these things comes under this molecular modeling. First of all, we have to model the molecules. You know, according to this computational any visualizers, if you want to open any molecules, can be a drug or can be a protein, it can be DNA or RNA, whatever biomolecule you want to open in a visualizer, like Discovery Studio. Okay, we assume that one atom, I mean, one particle is one atom. One particle is one atom of any molecule. Okay, this is what a basic assumptions we make. Bohr's atomic model. You remember whatever we have studied in chemistry. So that chemistry concepts, if you want to implement in the in silico studies, even it can be visualizing the any biomolecules. We use these kind of a concepts, whatever is studied in chemistry. So whatever the molecular modeling, majorly we study on the computational chemistry, and of course this computational chemistry we use it for drug design and other various application, can be even can be for vaccine design. So in molecular modeling, majorly we use the molecular mechanics approach. More about this we'll be discussing in the subsequent sessions. Now let me take you to the types of molecular modeling. As I earlier mentioned, molecular modeling can be a small molecule or it can be a macromolecule. So these are the two types we can see. Macromolecule we have seen in the earlier session. Now let me take you to the modeling of small molecules. How to model small molecules. So molecular mechanics, basically one should remember that. I mean, in molecular modeling, molecular mechanics we use, isn't it? We use, of course, we start from the classical mechanics to model molecular systems. What do you mean by that? Classical mechanics. In classical mechanics, generally we study the motion of the molecules, okay, through Newtonian's law of motion. So studying about Newtonian's law of motion is what we call it as classical mechanics. Okay, we start. This is the basic study. We use it in molecular mechanics. Later, we try to implement statistical mechanics and then quantum mechanics. Okay, and if you see this model, what model we use for the molecular mechanics, born Oppenheimer approximation. If you see in any tools, it can be Swiss model, it can be ITS, or it can be free offline tools for modeling the small molecules like drugs. So this is the concept we use. Okay, that is a born Oppenheimer approximation is used, and of course, we try to calculate even force fields for the atoms of a molecule. What is the difference between atom and molecule? Molecule is made up of multiple atoms, isn't it? These are interconnected, and of course, these are hold together through internal energy. But if there is a motion, if there is a dynamics, obviously we can find even Bernoulli's law of energy also is applicable. You can see the kinetic energy, potential energy. So this kind of energy we can able to see if the molecules are in motion. Okay, but based on what kind of a study we study. Okay, so based on that only we use this kind of a forces for calculation. Now let me take you to the atomistic molecular mechanic methods. I told you earlier, each atom is simulated as one particle. In any visualizer, it can be Discovery Studio visualizer, Python molecular viewer. Even in Autodoc, we have Python molecular viewer, isn't it? Even in there. So whatever visualizer as small, Swiss PDB viewer, everywhere we find each atom. One atom is one particle. So each particle is assigned a radius, typically a van der Waals radius, polarizability, and a constant net charge. Okay, and if you see the bonded interactions are treated as springs. Bonded interactions are treated as springs with an equilibrium distance equal to the experimental or calculated bond length. Okay, so the motions or generally all the atoms are not moving with the Brownian motion. Okay, many molecules, like especially a drug interacting with a protein, we observe this kind of a spring-like motion. Okay, and if you see this bonding and non-bonding interactions, I think this is repeated. So bond stretch. So actually, there are three types of bonded interactions. The first type of bonded interaction is bonded stretch. So in this bond stretch, this atom is going to. This is the initial length, isn't it? But if it is going to stretch, it can also stretch up to this position. Okay, so from here to this is the initial stage, but the final stage can also be up to this. If it is going to stretch, because this is a bond stretch. And for calculating the binding energy of the bond stretch, we can use this formula. Now let me take you to the next thing, bond bend. So in bond bend, this is the initial angle. This is the initial angle of the bond bend. But even you can able to see if this atoms can move up to this. This might be your final bond bend. So initially, you can able to see around 60 degree, more than 60, is it? No, 60 degree. But if it can able to move up to 120 degree, so then this is a change in angle in the bond bend. So the potential binding energy, bond bend energy can also be calculated like that. And this is the third bonding interaction, bond torsion. So it can be a molecular modeling, docking, simulation. We find all these bonded interactions, all these three types of bonded interactions in the atoms. Okay, obviously in the molecule you have so many atoms, isn't it? So in the molecule you observe like this kind of three bonded interactions. What do you mean by bond torsion? See, you have st- this kind of a functional group, isn't it? This will rotate. If it rotates like this, it can be a clockwise or anti-clockwise through axial rotation, axial direction. That's what we call it as a bond torsion. So for this, you can calculate the energy through this formula. So these three summation, totally we call it as bonded interactions, as I mentioned earlier. So in one molecule, you can find these three types of interactions also. And if you, whatever you sum, you obtain is the total bonded energy interaction. There is also a second type of non-bonded interaction, that is non-bonded interactions. In the non-bonded interactions, we have two types. One is van der Waals, the other is the electrostatic. The summation of both is what non-bonded interaction. And this is the formula for van der Waals and electrostatic uh, non-bonded interaction. The summation of bonded and non-bonded is the total energy of interaction. Okay. And of course, whatever energy we calculate, if you want to link with the classical mechanics, that is what Newtonian law of motion, you know that F equals ma. This is according to Newton's law. But how to calculate energy? If you want to calculate energy, so energy is calculated as force into distance. If you calculate the force, even through the distance of one atom to the another atom, you can able to calculate the energy. So this is the simple concept they have used to determine energy. So to make a protein more stable, we go for energy minimization protocol. So first we calculate energy and we do the energy minimization. Okay, so how the energy is calculated initially through this formula. Got it? So now I take you to the molecular modeling tools. So till now, whatever I discussed is related to chemistry and also physics. We linked chemistry and physics, and then we brought a computational part. If you want to visualize a protein molecule, if you are viewing a molecule, if you are rotating a molecule, or if you want to dock a ligand with a protein molecule, okay, in all the cases, we are using these kind of concepts. What concepts? This boundary interactions, non-boundary interactions. If you take a kind of a ligand receptor, I show you one kind of an example now. So I take you directly to the some talk to file. I have talked plenty of files. So one docked complex. Okay. Jaldanamycin TP53 complex. Jaldanamycin is the trap1 inhibitor. So I'm going to open a ligand receptor complex. In a ligand receptor complex, obviously we can also find similar kind of bonded and non-bonded interactions. If you want to see the 2D diagram, if I click this, this is my ligand, the Jaldanamycin ligand. So the Jaldanamycin ligand is interacting with the amino acids of the specific protein. And you can see these are the interactions of one atom to the another atom. A ligand, you have plenty of atoms. In Jaldanamycin, you have plenty of atoms. And similarly, in amino acids, you have plenty of atoms. Even leucine is made up of plenty of atoms in 15th position. In lysine in 17th position also have plenty of atoms. From chiral carbon atom to amino group, carboxyl group. You remember how the structure of amino acid looks like? Okay, so everywhere you can find this is the common concept we use for building. Now, let me take you to the our presentation. Molecular modeling tools for building 3D structures. So protein, Swiss modeler ITSL. So I told you Swiss modeler we are going to we use it for what kind of modeling? Homology modeling. For performing homology modeling, this is the best tool we can use. Easiest tool, and you can see the output is rapid. But if you want to work on threading in another concept, this is one of the best tool. Okay, what is that? ITSL. And for peptides, we can use through Vega F-Fold 3 server. Vega is a free tool. You can download and install, and through offline, it can work. It's an open source software. And F-Fold 3 is an online server. So in online server, you can able to feed the peptide sequence and build the peptides. For chemical compounds, you can able to build through ASD Chem Sketch and Pro TRG server. This is an online server, and this ASD Chem Sketch is an open source software. That is, you can download and install, and without internet also you can work with it. But if you integrate with internet, if you link with internet, you can also use other options. ASD ChemSketch, the offline tool, can also be linked with online, and we can also work with online applications also. Now I'll be showing all this, how to use the ASD ChemSketch. In ProDRG online server, we have limitations. In one day, okay, the one user can able to model only 3 molecules. So first you have to register, and after logging in, you can able to model only 3 molecules. Actually, you are provided with tokens. like how you go to the milk booth and collect the tokens. Similarly, here they provide three tokens. Only for three molecules can be modeled. So instead of going to the ProTRG, you can also go to the ASD Chem Sketch. But a few advantages are there in ProTRG. If you want to model the molecules in different formats, we have plenty of formats. More than 8 to 9 formats are available in ProTRG. So you can model using ProTRG. Or else you do one thing. You can model using the ASD Chem Sketch, and we can able to convert into 2 or 3 formats. Whatever formats we convert, we can able to convert the other formats through tools like OpenBabel. Use the tools like OpenBabel or use the tool like Babelwin. Okay, earlier we had OpenBabel as an online tool, but now we also have offline tools. Babelwin is a purely offline tool. It's open source software. We can also try with it, and we can go with the mole inspiration, an online tool for even for modeling the chemical compounds. RNA for mRNA server. Now, if you go with the tertiary structure prediction, okay, we use the, you remember, this is what we have seen in earlier classes, homology modeling, threading, and ab initio. These are the three methods. And you see for the tools present in these three methods are available in XPassive web server. Okay, so for tertiary or quaternary structure of prediction of macromolecules, we can use any of these three methods. Swiss model, you remember? I think I have also shown you the outputs and how to validate the stereochemical quality of a protein. This is also available in Swiss model itself. Ramachandran plot. ITaser. We have to register and we have to log in for modeling a protein molecules. PepFold 3. How a PepFold 3 looks like. A PepFold 3 server looks like this. Okay, simply in Google type for PepFold 3, you get a server like this. Simply click Run PepFold 3. If you click Run PepFold 3, you get a window like this. In this window, you have to type the amino acid sequence. Just like that, if you feed the amino acid sequence and if you click Run, which is present at the top, okay, you will be getting an output like this. Okay, we can also find in different styles. If you want to download, we can able to download through an option called model1.pdb. An option is there, isn't it? So try to download this pdb file. So similarly, if we want to download, work on tRNA structures, we can use mRNA server. And mole inspiration can also be used to draw the molecules to not to generate the 3D structure of molecules or to calculate the properties and bioactivities. ACD Chem Sketch. This is an offline tool, so the anyone can download and install. Okay, no price is there. It's not a commercial software. So this is a good software for chemical for modeling the chemical molecules. Of course, whatever we work on the small molecules, we call this as chem informatics. There is a branch of study called chem informatics. What is chem informatics? The chem informatics is modeling these small molecules. Okay, modeling the small molecules and using the small molecules for determining activity and other properties. That's what we call it as a chem informatics. Okay, bioinformatics. Can I ask you a few questions? From beginning, I am not asking you any questions. I ask you very simple questions. So mention me what is the difference between bioinformatics and chem informatics. Just now I explained you what is. Send us this PDF, madam. It's available, madam. You can this video. I am sending it to you, isn't it? I am sending you the video. Only thing is that you have to practice and you can form the slides. Okay, so I'll be sending you all the videos for sure on the same day or on the next day morning. Okay, if you have any doubts, you can ask me. Sir, can you also add a point on advantages and limitations of using AlphaFold? Okay, so many questions. First, I can answer your questions. And also ask you one question. Difference between bioinformatics and chemoinformatics. The participants can also try this. And I can, meanwhile, I can check the questions of the participants and answer. Can we target where from here? Can we target the interacting protein as disease target, sir? Interacting proteins as disease. Yes, yes, you're right. That's what I mentioned you from the beginning. That's what system biology approach. Uh, Two or more atoms together is a molecule. Yes, you're right. advantages and limitations of using a phone. If you type in chat GPT, you will be getting plenty of information on advantages and limitations. Okay, so if I take on more theory part, so we limit the hands-on skills whatever I provide. I think I remember that you requested me to take more on the databases, go to the browser and explore on the data. So that's what I am committing at present. So I think you are the participant. You asked me to do like that. Okay. So AlphaFold. Anyhow, you asked me, so I'm answering for you. AlphaFold is an AI-enabled 3D structure of protein database. AI. AI. It means that we are integrating ML, machine learning techniques. If you are using a machine learning techniques, and if you are going with a specific machine learning technique, for example, if you see proof for protein interaction, the most widely used machine learning technique is support vector machine. And for protein prediction, we use neural networks. Other than neural networks, we can use hidden Markov models and other information also. But which is best? That what I am mentioning you. For protein interaction, better go with the SVM, and also use the Bayesian model. SVM, when SVM is integrated with the Bayesian model. Okay, the machine learning techniques, we are not going to use a single machine learning technique. We are integrating different machine learning techniques, and we can also check the accuracy and the prediction accuracy and other parameters in the machine learning techniques. Got it? So whatever we predict in AlphaFold is not 100% precise when compared to wet lab techniques like X-ray crystallography, NMR, whatever we do in our research lab. So these are the limitations. The limitations we can predict, it can be 80% accuracy or it can be 90% accuracy, but it can't be 100% accuracy. The accuracy what we get is only from the wet lab techniques. Only we have to work on this X-ray or NMR. Okay. AlphaFold is simply a machine learning kind of a technique. Of course, this is an AI-enabled kind of a database, a 3D protein structure database. I believe now I answered your answer, madam. Now, ProTLG is working now on online mode. Yes, you are right. It works on online mode only. Guys, can you explain how to use ProDRG? Simply, I have to go to the ProDRG. See, that's what I'm going to explain you now. Okay, so I'm going to close all this. So, how to work on this kind of a small molecule servers? So, step by step, I can come. So, I didn't even complete my presentation. After completing my presentation, I take you to this kind of a tools. ASD Chem Sketch can also use. No, this is not a ASD Chem Sketch. This is a Vega. There is a tool called Vega. This is also a free tool. So this free tool can also be used to draw the peptides. Simply you have to feed the amino acids. So for feeding the amino acids, we simply have to click these buttons. So initially I clicked leucine. Okay, L stands for leucine, V stands for valine, aspargine. So simply you have to click these buttons in the Vega tool. And if you click this tool, you can able to get a 3D structure. You click build and you can build in the form of .pdb. And also you can see the structure type, what kind of a structure it is going to form. So this information also you can able to find it from here. And that's it. Now let me take you through each and every tool. First, you want me to take you directly to the ProDRG. So I show you now. So first, I'm taking you to the PepFold 3, which is most important. I'm showing that giving you the priority. We just have 6 more minutes to complete. So PepFold 3. So I'm taking you to the PepFold 3. So this is an online tool. So we have run PepFold 3. Can I click this run PepFold 3? So even if you have a few more questions, I can drop the same in the chat box. So while after completion, I can check your questions and answer for it. If my answers are not clear, you can still ask me by unmuting and ask me so that I can clear your answers. Okay, I can clarify your. And now let me take you to the peptides. For example, if I want to type an amino acid sequence. Generally, how you can get an peptide amino acid sequence? If you go for epitope prediction for uh, viral vaccines. Okay, obviously we get these kind of an amino acid peptides from 8 amino acids to 15 amino acids. So now I give randomly some 9 amino acid peptide sequence. So before that, I just asked you a few questions, but whether you answered this or not. What is the difference between cheminformatics and bioinformatics? Cheminformatics designs molecules to hit targets that bioinformatics identifies. Okay, trying well, but we are making more complex. How do we target the nucleotide like ligands on genetic therapy? Is there any tools? This is more advanced. More advanced. Of course, we can use all these computer drug design techniques for this. Even we can able to use the techniques. Actually, we are half through. Half knowledge is always dangerous. If only if we complete perfectly the drug design and molecular mechanics, then if you ask these questions, then I can whatever I explain, definitely it reaches your mind. Now, partially your partial knowledge you have acquired. Okay, let me complete up to 10 sessions. So for the answering this question, you have to complete 10 sessions. You know. Okay, focus on chemical data. Very good. It's very simple answers. Renika Devi is providing very good answers. Cheminformatics focusing on chemical data. Bioinformatics focus on biological data. For example, protein data. That's it. Simple answers. This is the exact answer I am anticipating from the participants. Okay. So cheminformatics, small chemical molecules. Bioinformatics, macromolecule like protein sequences, protein structures, etc. Sequence structures, everything. All our data only. Isn't it? Good. Now, let me. I was explaining you something else. Peptide. Okay. So there are different technical similar terms. Peptide, polypeptide, and then protein. What is the difference between this? You know, Peptide, polypeptide, proteins, lipopeptides. We also get one more new thing. See, all are looking very similar. But can you tell me what is this difference between this? If you see peptide is also made up of amino acids. The building blocks are amino acids. Similarly, polypeptide. However, you find for protein, the same definition applies for all the molecules. Except the lipopeptides, you have lipids linked with the peptides. So lipids are also connected. Peptides are small amino acids. Good, but there should be a range, isn't it? So only through range we can distinguish all these three. So around up to 20, whatever we have is a peptide. Generally, a peptide is not less than 8 amino acid for many therapeutic purposes. So minimum 8 you can able to observe. Okay, so 8 to 20 is the range for peptide. And if you see this polypeptide, 50 to 75. Polypeptide is 50 to 75, and above 75 is obviously a protein. A smallest protein sequence we work on is a insulin ligand. The insulin ligand is around 110 amino acid. The smallest protein what we can able to work is an insulin ligand. Okay, that is a human insulin ligand. I'm telling about the human proteins. Insulin receptor is large in size, more than 700 amino acids. Okay. Now let me take you in. So I was explaining you about the peptide, and I was designing about the peptide. Can I give you something random amino acid and try to build a kind of a peptide? We have very less time. LDK, N. Whether we have O, we don't have O, isn't it? But other things we have. Okay. Now I just type few amino acids, and randomly whatever I type, I'm going to click run. So it's asking for no, don't give the caps or however they mentioned, provide that. Click OK. Now I'll be getting a peptide soon. Now, meanwhile, I can take you to the ASDCAM stage. I have already installed, I believe. Let me take you to the thing. If you have Vega, we can also build the peptide. ACD. Yes, I have this. Software is Sketch. If you install this ACD Sketch, the window looks like this. See, now initially, if you want to draw this molecule, you can also draw the molecule. Just drag and drop. Okay, if you want to draw, have a kind of a bond, okay, no, something happened. Uh, I take this, okay, control is it, control is it. I take this and I'm going to stitch S, OH, and this, can I make it as COH? OH, OH. But how to make COH? CH3 OH. So what I have to make? I have to make COH. Then I have to click. If how I am making, So if I want to have a CH3, this is a CH3. If I click here, this is CH3. Okay, if I want to click here, now the CH3 is converted. But if I want to convert as OH, I'm converting like this. So now I have created a benzene molecule and created just a CH3 and OH. If I want to find what molecule is this, how to find, even we can have a finder here, PubChem. So if I want to search exact structure from the PubChem, there is an icon here. I click, you got this information from the browser, then you click OK. So whatever simple molecule I have drawn, okay, now it's going to search in PubChem and it's going to tell what molecule is this. This is orthocresol. Whatever molecule I have drawn is orthocresol. If you want to find the properties of the orthocresol, you can able to click inside and you find everything in the PubChem. NCBI PubChem, you can find these properties. Similarly, okay, we can also search in E molecules. We can search in ChemSpider. These are the other databases. If you want to determine the properties, we also have this icon log p. If I click this log p, immediately you got the value. See, even you got a mean plus or minus standard deviation. The log p value is 1.94 plus or minus 0.19. Okay, this is the information. And if you want to draw even the other elements in the periodic table, we also have the periodic table also. Okay, so can I show you the periodic table? So see here, and we can also see the functional groups. We can also have the amino acids, so we can draw the amino acids also. Okay, so a table of radicals, we have C3, C4, we have the 6-member ring, cyclobutane ring, cyclopropane ring. If you want to draw any of these structures and you want to use any of these functional groups, we can use this. Okay, and even any of the elements in the periodic table, bromine, potassium, chlorine. If you want to incorporate anything new, you click this new, any, you can give something, armenium, rubidium. But generally, this won't, we won't use it for chem informatics. We have chlorine in the protein. We have phosphorus. So these are already present here. So you can drag and drop and you can work with it. Got it? So one thing you have to remember, so we can able to use this to convert it as 2D structures, 3D structures. We can find the exact structure of the molecule. We can find the substructures of the molecule. Okay, through different databases, we can determine the physico-chemical properties. So this is an extensive, excellent database, a chem informatics database. Okay, and if you want to model small molecules, this is the tool we can try. And we can use Vega for protein peptide. So you see, in ProDRG, we have got the result. So in the online tool, just now we have tried, isn't it? If you want to download this pepfold, the model of the peptide, click this download. So you get in this browser. It's just like that, you could just copy and paste it in a notepad and open it in a visualizer. If you open it in a visualizer, it is going to open. Okay, so I'm just opening a new notepad actually. So in notepad, actually I'm creating a new tab and then pasting this and file, save as, While saving, you save it as .pdb. So from beginning, I am saving it in desktop, bioinformatics demo in CADVD. I'm going to save this as a peptide.pdb. So peptide.pdb should be in double quotes. Peptide.pdb should be in double quotes. So default it's in text documents and saved it in peptide.pdb. So this you can able to open it in Visualize Select Discovery Studio. So I think we already taken more than 5 minutes on today. Okay, so I have completed how to model the peptide and how to model the small molecule and how to download and how to open. Okay, so if anything is missed in this class, we'll be continue reading in the next sessions. Okay, for whatever questions you ask, we'll be seeing the answers in the forthcoming sessions. If I complete that sessions completely, then you get a knowledge on that. Okay, and you can ask questions accordingly. But very good interaction in this class, many participants are raising the questions whatever you have in your mind, having different activities like anti-cancer. Okay, I don't understand this question, but I have completed my session. If you have any doubts, you can ask me. For the others, thank you for interaction and patience. Any doubts you have? Okay, thank you. If no doubts, we can see on tomorrow. Have a great weekend to you all. Bye.